Welcome to our lecture discussion of the muscular system. We're looking in chapter of your textbook. If you open up your textbook, we are in chapter 6 of the textbook. So don't forget to do the lecture homework that is on the Mastering A&P website to prepare for the lecture exam coming up next week on the skeletal and muscular systems. So be sure to have done the lecture homework for both the skeletal system for chapter 5 and the muscular system for chapter 6 to help prepare you for the lecture exam next week. So starting with the muscular system, looking at the functions that are unique to the muscular system. Body movement. We know that muscles attach to the skeleton, to the bones, to pull the bones at joints and create movement. So that's a very important function of the muscular system. Maintaining posture, we have muscles of the back and muscles of the abdomen that help hold us upright when we're walking. Respiration, we have the diaphragm, which is found as a border between the thoracic and abdominal cavities. The lungs rest on the diaphragm, and when we contract the diaphragm, when we inhale, it causes the diaphragm to move lower into the abdominal, toward the abdominal region, and that allows the lungs to inflate as they fill with air. And then when we exhale, the diaphragm relaxes and it curves upward again into the thoracic cavity and again allows the lungs to deflate as we exhale air. And that diaphragm is a very, very important muscle for respiration. And it's not under voluntary control, so it's one of those exceptions that most of skeletal muscle is under voluntary control, but it is not under, the diaphragm is not under voluntary control because we know that we can breathe while we're sleeping. Production of body heat. This is a really important function that not most people think about or realize, but we know that the muscles, when they contract, they produce energy, and some of that energy is given off as heat. So even our just basic muscle tone that we have for maintaining posture and just in general muscle tone, as we have tone in our blood vessels, the smooth muscle, all of that is using energy, and some of that is given off as waste heat. So we have our 98.6 degree Fahrenheit body temperature is a large result of muscle tone and muscle contraction. And as you know, when you exercise and you have more than normal muscle contraction, that does produce a lot of heat. Communication. Muscle cells communicate with one another, especially if we look in the heart. The muscle cells of the heart contract as a unit in the ventricles, and also we see this in the atria to propel blood throughout the heart and then out of the heart to the rest of the body. We have smooth muscle that lines our organs such as the bladder, the stomach, the intestines, the uterus. Those are all organs that constrict as a result of smooth muscle contraction, and that helps to propel substances throughout the body. The same thing with our blood vessels. Our blood vessels are lined with smooth muscle, and when they contract, that can increase blood pressure to speed blood flow to, for example, working muscles during exercise, or what else? To the brain, if we get up from a lying down position, the blood vessels of the carotid arteries will constrict to allow blood flow to increase to the brain. Also, kind of on the opposite side of that, we do see dilation of blood vessels when we want increased blood flow. So, for example, when you're exercising, the blood vessels will dilate in the muscles, to skeletal muscles, to cause increased blood flow. And then the blood vessels to for example, the digestive organs would constrict during exercise because it's not a priority to digest food when we're exercising. And that's why some people might get a stomach ache if they exercise too soon after a meal. So we maintain blood pressure primarily by dilating and constricting the blood vessels. So we'll get more into that when we get into the cardiovascular system. With the heartbeat, that's a perfect example of synchronized muscle contraction. When we get into the heart, the cardiovascular system, we'll talk about how the heart has special cells, muscle cells, that can generate their own impulse and cause contraction without interaction with the nervous system. But oftentimes the heartbeat is tightly controlled by the nervous system but we'll get into detail on that when we study the cardiovascular system. So unique properties of muscle tissue, if we look at it as a tissue, we find that muscle tissue is contractile. It exhibits contractility, which means that muscle can shorten. So when the biceps shorten, for example, it pulls the forearm toward the shoulder, and that is an example of a shortening muscle, the biceps muscle shortening when we flex our arm. Excitability means that muscle cells respond to some stimulus, and typically it's a nervous stimulus. When we think about a motion that we want to make, our brain generates an impulse that travels to that muscle and causes that muscle to contract, so that muscle can respond to a stimulus. Oftentimes we have reflexes that are the stimulus, so it may not be a thought, it might be some environmental stimulus that causes a reflex to occur, like for example when we shine a light into the pupil of the eye, it causes the muscles of the iris to constrict the pupil and make it smaller. If we tap on the patellar ligament, that causes that mechanical impulse to travel to the quadriceps muscles and causing that to kick the knee outward. So those are examples of excitability. Extensibility means our muscles can be stretched beyond a resting length. We know when we sit down and stretch our legs, we can bend over toward our toes, a little closer to our toes than if we were just sitting normally, and we don't tear our muscles because they can be stretched within a reasonable range. We know we can tear muscles also if we go beyond that resting, if we go beyond the reasonable extensibility range. And we know a good stretching program throughout life keeps our muscles extensible, because if we don't, over time our muscles become tighter and tighter, and that can cause problems with mobility. Elasticity, our muscles are elastic, that if we do stretch them, they will bounce back to a resting length. For example, if you rest your arm, just let it hang loosely on the side of your body, it has a slightly bent position, but you're able to straighten that. That's the extensibility characteristic. And then if you relax your arm again, you'll notice that it goes back to a slightly bent position. And that's a demonstration of elasticity, that the muscle can rebound or return to its original resting length after we stretch it. So be sure for the lecture exam that you understand the definitions of each of the terms: contractility, excitability, extensibility, and elasticity. There's 3 different types of tissues in the body, muscle tissue that is, and that text page reference is not correct, so I would have you change that, and the descriptions of the 3 different types of muscle tissue is actually on page 182 in your textbook. So I'm going to change that right now. It's page 182, and we'll continue where we were. So the 3 different muscle tissue types: skeletal muscle is voluntary. It has these striped striations on it, so we can see here those vertical stripes are the striations. Notice that each cell is very long and cylindrical in skeletal muscle, and the nuclei, there's more than one per muscle cell, and they're near the edge of the cell. So that's very characteristic of skeletal muscle. We're looking for striations, long cylindrical cells, and multiple nuclei. If we look at the other types of muscle tissue that's shown in your textbook on page 182, and we also looked at these in lab already, so I'm not going to revisit these all again, but smooth muscle is involuntary. That's what allows our food to move through our digestive tract, allows urine to move out of the bladder, out of the body. All of that is smooth muscle, and that is not under voluntary control. And those muscle cells we looked at in lab were tapered on the ends, and they have a single nucleus and no striations. Cardiac muscle we also looked at in lab. That's the muscle cells that make up the heart. They're very similar to skeletal muscle in that they're striated, but like smooth muscle, they are involuntary muscle cells, and they have a single nucleus. So skeletal muscle structure, if we look at skeletal muscle throughout the lifespan, once we're done growing, the number of muscle cells we have in our muscles remains constant, which is the same for adipose tissue. The number of fat cells that we have at the end of childhood remain constant. So when we gain or lose weight, we're not increasing the numbers of cells, we are increasing the size of adipose cells. The same thing with muscle cells. If we exercise or weight lift, we're increasing the size of those muscle cells. We're not increasing the number of muscle cells. Looking at the microscopic anatomy of a muscle is quite detailed, and page 183 in your textbook has a nice diagram that matches what we're looking at here, and also page 186 goes into a little more detail as well. But we notice here that we have the outer covering of muscle is called the epimysium. So that's an outer connective tissue wrapping around muscle, and when those muscle fibers end and we have just epimysium connecting to bone, we call that portion the tendon. So a tendon is a connective tissue that connects muscle to bone. Another connective tissue wrapping we see that in muscle are these bundles of muscle fibers here called fascicles. This is a fascicle. Here's another fascicle. Here's another fascicle. The connective tissue that we find around the fascicles inside of a muscle is called perimysium. So this is perimysium. And this is a fascicle. It's a bundle of muscle fibers. Again, this outer covering would be called perimysium. And then within each fascicle are the muscle fibers. So here's one muscle fiber that's been pulled out, but there's a number of them we can see here in this fascicle. And the connective tissue between the muscle fibers within a fascicle is called endomysium. So we have three different connective tissue separations within muscle. We have the epimysium on the outside of the entire muscle. We have the perimysium surrounding each fascicle. And we have endomysium inside a fascicle separating the individual muscle cells. And recall that the muscle cell itself has a tissue covering which is just simply the plasma membrane. So that's not, we wouldn't even classify that as a tissue. Actually, I should correct myself on that because it's not made up of individual cells. The plasma membrane is made up of proteins, carbohydrates, phospholipids. So that is the plasma membrane that covers the muscle fiber. So if we look at what is inside, deep inside of a muscle fiber, we have two myofilaments called actin and myosin. And this is shown on page 186 in your textbook. If we look at a myofibril, which is shown here, I'm going to jump over to a picture here. Actually, we're going to go in Blackboard. Under participation assignments, you will find a worksheet talking about microscopic structure of muscle. So we're going to look at that here, and I would recommend that you fill this out as a group. So if we look at, with this we just filled out together, or just looked at together, this would be the tendon, what you would put here. This would be epimysium here. This would be endomysium here. This would be, now remember, this is a muscle cell. I'm sorry, muscle fascicle. So this would be an individual muscle cell. So muscle cell or fiber would go here. This also is endomysium. This is muscle fascicle. And what makes up that muscle fascicle is perimysium. So this too would be fascicle or perimysium. It's the outer covering. And then we have bone. So this next diagram on the bottom here is looking at an individual muscle fiber. So it's taking this structure here and blowing it up and looking at it in more detail. So that's what we're seeing here. So in a muscle fiber, what we see is even smaller units within a muscle fiber, which are called myofibrils. So that's what goes here, myofibril. And then within that myofibril is an even smaller unit called myofilaments. And that's what will go here. So we have these small, long units within a muscle fiber called myofibrils, which are made up of even smaller units called myofilaments. So this diagram here we would label as a nucleus. And this banded pattern, we have repeating light and dark bands. We looked at that in muscle tissue. Those are the striations that we've already looked at. So this is striations. So if we zero in on a myofibril, we'll see that it has a structure like this. We have repeating units end to end, making the entire length of the myofibril look like this if we could zero in on it with a microscope. And this again is shown on page 186, and figure B shows what that myofibril looks like up close. So we have two myofilaments that make up the myofibril. We have these blue lines, which are called the actin myofilaments, and we have the red lines, which are the myosin myofilaments. This region of a myofibril where we have overlapping actin and myosin myofilaments is called a sarcomere. And the border of a sarcomere are at these points where the actin myofilament is tethered to the myofibril, and these regions here are called Z-discs. So from one Z-disc to the next adjacent Z-disc, we call that, again, a sarcomere. So that's what would go here, is sarcomere. You could ignore the rest of these terms, because this is from the other diagram. So I'll just fix that now while I'm on here. Okay, so let's go back to the PowerPoint, and a lot of this is best understood by looking at an animation. So we'll start with this and watch the animation here.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
Our course with the A band, H zone, I band, don't worry about that, but do focus on, here's the Z disc we talked about, or Z line is another name for it, where the actin is tethered down to the myofibril. So here we can see there's one Z line here, there's one Z line here, and that distance from point to point is called a sarcomere, and that's labeled here. Notice there's space here where actin and myosin are not overlapping. So when we contract muscle, what happens is the amount of overlap is going to increase as myosin pulls on actin and pulls it toward the center of the myofibril, or the center of the sarcomere. So we can see these myosins are like oars on a boat, pulling on the water and moving it forward to the center. And muscle contraction stops when those two actin myofilaments are touching each other. So if we answer the quiz here, we can say muscle contraction is caused by looking at our choices. We have, this is myelin, that's not even what we're talking about myosin, is we have actin filaments sliding past myofilaments. So we can see the actin is sliding past the myosin. But if we look at it, we'll notice that neither are decreasing in their length. Myosin doesn't get shorter, actin doesn't get shorter, just the amount of overlap increases. So the sarcomere does get shorter, because if I measure this distance from here to here throughout this muscle contraction, watch what happens. Doesn't that line get shorter for the sarcomere? So the sarcomere shortens, but the actin and the myosin, actin and myosin remain the same. And that's a real key concept for studying muscle contraction. So we call this process of actin and myosin interacting, we call that the sliding filament model, because it's actin sliding over myosin to shorten the sarcomeres. And that's a real key concept, so I would underline and highlight so you know what's shortening here. It's the sarcomere, but it's not actin or myosin, they're just sliding over each other. So actin and myosin do not change length. So when we relax the muscle, what happens is myosin detaches from actin, and we said that muscle is elastic, so it returns back to its resting length again for another stimulus to cause myosin and actin to interact again. So again, we're looking at the distance from one Z-disc to another. That defines a sarcomere. So here we can see a sarcomere is defined. Here's another sarcomere. And we're looking at the actin and the myosin interacting with one another. So I have another one. I think this is a different one. We'll take a look. If it's the same one, I won't show it again. Yeah, it's the same one. Okay. So we'll just get rid of that one.
In a relaxed muscle, actin and myosin myofilaments lie side by side.
Again, the point being that actin and myosin are sliding over one another. And which myofilament is doing all the work? That's the big concept. The cell that's, or the myofilament that's doing all the work here is the myosin. That requires ATP in order for that myosin to pull on that actin and pull it inward to shorten that myofibril. And if we shorten the myofibril, we shorten the whole muscle cell, which shortens the fascicle, which shortens the muscle. So these tiny little units that are so small doing the work of contraction, that work is transmitted to the entire muscle. So how can we tell if we have the energy to lift, say, a 10-pound weight versus a 100-pound weight? Well, it depends upon the number of muscle cells that are served by a particular nerve, and that's what we call a motor unit. It's the muscle fiber supplied by a single motor fiber. So here we can see there's two motor units. There's one that's serving these muscle cells within the fascicle, the one shown in green, and the other motor unit is serving these muscle cells. Notice it's within the same muscle. We have different nerves supplying muscle cells in the same muscle. So the more motor units we recruit to an activity, the stronger that muscle contraction. And we're never sure exactly how strong our muscles are, because we find that when people are under great amounts of stress, they can do amazing things in terms of muscle contraction. And let's watch a little video that demonstrates that.
The Sandia Mountains in New Mexico are a challenge for climbers. The granite faces are notoriously unstable.
It's a little steep here.
Mark was in front, and I lost my footing just a little, and by instinct to catch myself, I reached over and put my hands on the wall. And that's when all hell broke loose. The wall essentially came off in my hands.
This thing was huge. And how it didn't crush him to death, I don't know, but I just watched him and I couldn't believe he was actually still alive.
St. John is trapped underneath. The slab of rock is heavy enough to crush his ribs, but he's just able to use his arm muscles to hold it off. What's worse, he's landed on a sloping ledge. He's now sliding toward a high cliff and certain death. He's stunned and in shock, but his body has already shifted into overdrive. Any chance St. John has of survival depends on what's locked in the muscles of his arms, chest, and shoulders. But how can his muscles move something so massive? Muscle tissue works by contracting, pulling on the bone, using it like a lever. These contractions take place at a microscopic level. Each of St. John's muscles is made up of thousands of individual fibers, bundled together like the wires of cables. Though they may get bigger or smaller through life, we're born with every muscle fiber we'll ever have. Within each fiber are yet smaller filaments. To activate the muscle, chemical triggers cause filaments next to each other to ratchet together, intermeshing like locking fingers. As they slide past each other, the whole muscle fiber gets shorter. These contractions are responsible for all our muscle movement. Even when exerting ourselves, most of us only use about a third of the fibers in our muscles at any one time. But if St. John's to live, with the cliff edge fast approaching, he will have to heave a rock weighing more than half a ton. In such extreme danger, he, like the rest of us, is able to do something that normally would be impossible. He's unleashed all the power in his muscles. St. John heaved a rock weighing 1,200
how
pounds.
strong our muscles can be if we recruit enough motor units to that activity. We've heard examples of this where you have a mother who frantically finds her young child trapped under the car in a car accident. She's able to lift the car off of her child. So it's hard to really know how strong our muscles are, because to try to create those stressful conditions in a laboratory to do testing is impossible. It's only when we have huge amounts of adrenaline that we can see these incredible feats of muscle contraction and strength. So that leads us to, how does our muscle respond to a stimulus? And this is the result of an interaction between a neuron, which is a nerve cell, and the muscle cell. This is called the Neuromuscular Junction. And if we go back to our worksheet that I was showing you earlier, there's some things to label on here at the Neuromuscular Junction. And the first part, what you would label up here, is the motor neuron. And I should add that as a choice because it's not on there currently. I'm going to change this for next time. But this is the motor neuron, so that's a nerve cell. And the end of a motor neuron is called the axon terminal. So this answer here would be axon terminal. And that stimulates the muscle fiber. So here's the muscle cell itself. And here is the sarcolemma, which is the plasma membrane of a muscle cell. So this is the sarcolemma, which is the plasma membrane. And where that motor neuron interacts with the muscle cell, we find there's an indentation of the plasma membrane here, which is called the motor end plate. It's still the sarcolemma, but the special region underneath the motor neuron is called the motor end plate. So here you could put sarcolemma in parentheses and then give it the name motor end plate, because it's that region of the sarcolemma where the motor neuron interacts with the muscle cell. So there's a space between the motor neuron and the muscle cell membrane, and this space is called the synaptic cleft. And we're going to watch an animation that shows what happens in the synaptic cleft that transfers the stimulus from the neuron to the muscle cell. So this is synaptic cleft. Inside the motor neuron, we find these vesicles that contain a special chemical called acetylcholine. So you could write acetylcholine here, or ACH. And that's a special stimulating chemical that's going to stimulate this motor neuron. And then the sarcolemma has these tubules that go down deep into the muscle cell. So anything that's happening on the surface can be transmitted deep inside the muscle cell through these, it's almost like a sewer system down deep under the streets of a city. These are called T-tubules. So this right here you want to label T-tubule. And closely associated with the T-tubules and the actin and myosin filaments are a special smooth endoplasmic reticulum, which are called the sarcoplasmic reticulum. So that's what would go here, sarcoplasmic reticulum. And it's just basically a specialized smooth ER, and its job is to store extra calcium. Because when we study the skeletal system, we talked about how our body needs calcium for muscle contraction. And we're going to see that calcium is going to, when there's a stimulus, calcium leaves these smooth ERs, this sarcoplasmic reticulum, and binds to actin in order for myosin and actin to interact and cause muscle contraction. So calcium is really essential for muscle contraction because it allows myosin to bind to actin. Because normally actin is covered and unable to bind myosin unless calcium binds to it. And we'll watch an animation of that in a little bit as well. So going back to our PowerPoint slide, again looking at the parts, we have the sarcolemma. We have, they call this the postsynaptic membrane, but it's the same thing as the motor end plate. We have the synaptic cleft. We have vesicles, which are just membrane-bound sacs, but what they contain is acetylcholine. And what they're calling the presynaptic terminal, another name for that, is just the axon terminal. So here's our muscle cell with the myofibrils inside. So we're trying to get that stimulus from the outside of the muscle cell down deep to these myofibrils to get the actin and myosin to interact. So we'll watch an animation here on how that happens.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
But essentially we have a nerve impulse that travels down the motor neuron, and there is a special channel for calcium that is found outside the cells as well to come into the motor neuron. So there's calcium involved at two different levels. We have calcium coming in from the extracellular fluid into the neuron, and the role of calcium at that level is to stimulate these vesicles with acetylcholine to dump that acetylcholine into the synaptic cleft. So if we watch this, we'll see once calcium enters in from outside the motor neuron, it stimulates those vesicles to dump acetylcholine out into this space, the synaptic cleft, between the motor neuron and the muscle cell. When that is released, it binds to special channels here for sodium. So these are sodium channels here, where the key to opening those sodium channels is for acetylcholine to bind. And that causes sodium floating around in the extracellular fluid out here to enter the muscle cell. And we're going to learn when we study the nervous system that when sodium enters into any cell, if enough sodium enters, it stimulates an electrical impulse, and it stimulates that muscle cell. And here we see there's an enzyme present on the muscle cell membrane that can break down acetylcholine when it's no longer being used, so we can recycle it into its two basic parts and reassemble acetylcholine in that motor neuron. So these blue things here are just enzymes that break down acetylcholine. Because if we didn't break it down, it would constantly keep these channels open and sodium would continue to flow into the cell, continuing to stimulate this membrane. And that's not what we want. We want, periods of muscle contraction and relaxation. So going back to our worksheet here, once we stimulate that membrane, once it's at the surface, that impulse from the motor neuron, that impulse travels down these T tubules, remember, to stimulate release of calcium from these special smooth ERs called the sarcoplasmic reticulum. And that is the next step in this process of stimulating the muscle cell. So we'll watch that process now. So I'm going to turn the audio off and just talk through this one together. So here comes that stimulus over the surface of the muscle cell that came from the motor neuron as a result of the neuromuscular junction, and now it's going to go down the T tubules. So this is a real simplified view of the T tubules, but that's the indentations of that plasma membrane. And the yellow is the smooth ER, like I said, that stores calcium, called the sarcoplasmic reticulum. That's what the yellow is again. So the impulse comes down, it stimulates calcium to be released from those sarcoplasmic reticulums. Just watch it happen a couple times here. It's going to show calcium release here pretty soon. There's the calcium, and that calcium is going to come down and bind to actin. And when the calcium binds to the actin, that starts the interaction between actin and myosin. It actually is going to change the shape of the actin myofilament. Here's actin. Don't worry about the term tropomyosin for our purposes. You'll learn more about that in general A&P. But here's the actin myofilament, and when calcium binds, we're going to see how it changes the actin myofilament and exposes a binding site for myosin, which is sitting here waiting to interact with actin. There's the binding site there, and myosin can bind with actin, and with the help of ATP, pull that actin inward. And that process will continue until no more calcium or ATP is available. So what's actually working hard here in this relationship of actin and myosin, it's the myosin. The myosin is what requires ATP to get the head pointing in the right direction to attach to myosin, and then pull that, it's attached to actin, and to pull that actin toward the center of the myofibril or the center of the sarcomere. So the myosin requires the energy in the form of ATP. So it's kind of like a boat rowing in the water. The rower, the oars are what's doing all the work. So that's what the myosin head is doing. It's using ATP energy to bind to actin, and we're going to learn when we get into general A&P that ATP actually binds at the end of the power stroke here, causing the release of myosin from actin. But we'll study that in more detail when we get into general A&P. So moving back to our PowerPoint then, here we can see the sarcoplasmic reticulum shown in light blue here that stores the calcium and releases that calcium to the cytoplasm inside the muscle cell, because this is all a muscle cell, and interacts with those actin and myosin myofilaments. So, how does this apply to everyday life? Well, with exercise, our body responds to the demands placed on it. So if we exercise, say go a long distance with running or biking or swimming, for example, our muscles need more oxygen, more glucose to provide energy for that exercise. So as a result of endurance training, our muscle cells needing more oxygen, we will grow more blood vessels to that muscle to give it the blood and the oxygen and nutrients that it needs to do that activity. So when we train for, say, a marathon, our muscle cells in our legs will have more capillaries than someone who did not train for that activity. Our muscle cells will also have more mitochondria to take the oxygen from the blood and convert it into ATP. So we'll definitely see more mitochondria in those muscle cells to provide the energy for those working muscles. And there's also a special pigment found in muscle cells that helps us create to use energy more efficiently, and that's called myoglobin. So you'll also see more myoglobin in those cells as well. And we also see myoglobin in the meat that we eat. For example, chicken, where there's dark meat and white meat. Dark meat we're going to find in those parts of the chicken that have lots of myoglobin, because myoglobin is what makes dark meat dark meat. It also has more fat in those areas also, because those muscle cells use a lot of energy. So think of a chicken and what they do well. Chickens are good at running. So their legs and thigh muscles are dark meat because of a lot of myoglobin production. We would also find more mitochondria, more blood vessels, and more fat in those muscles because they're hard-working muscles. But the breast of the chicken is light. It's white meat, less myoglobin production, less mitochondria and capillaries and fat. because we don't see flocks of chickens flying overhead. But a duck, for example, ducks paddle around in the water. They use their legs quite a bit, and they also use their breast muscles for flying over long distances. And you'll find that duck meat is dark meat throughout. It doesn't have any white meat because all of the muscles are used, and it will have lots of mitochondria, myoglobin, capillaries, and fat for those long endurance exercises that ducks use, either paddling through a lake or a pond or flying over long distances. Resistance training, on the other hand, is when we're lifting weights. So the same things are going to happen. We're going to see more capillaries, more mitochondria, but we'll also find increased numbers of myofilaments and myofibrils, because when we exercise, we actually damage our muscle fibers, and the body responds by repairing those those tiny tears in those muscle fibers by creating more myofilaments, more myofibrils to again respond to the demands placed on that muscle. So we find people that are bodybuilders, their muscles are not only better for the exercise, but they also, better equipped for exercise with more capillaries and mitochondria, but also they're larger in size as well. Look at a typical cross-country runner. Maybe they can go a very long distance. They're not very bulky individuals. They don't have very bulky legs, but they do have muscles that are very efficient on creating ATP energy with the help of mitochondria. So as we age, what happens to our muscles over time? Well, we are less efficient at producing more blood vessels and more actin and myosin, myofilaments with exercise, so we see reduced muscle mass over time. We also find that at the level of the neuromuscular junction, our muscle cells don't respond as quickly to nervous stimulation when a neuron stimulates a muscle. So as a result, there's a more likely incidence of falls with the elderly, because if they are tripping over a cord or tripping on a rug at home, they can't move their legs quickly to support their feet and maintain their balance. They're more likely to fall. And because of less capillaries, less ATP usage, efficiency, we see reduced stamina. People can't go quite as long. They can't run as far as they used to. And it takes longer for those muscle cells to recover to be able to contract again. So there's longer rest periods needed between activities as we age. And as we have reduced muscle mass, that's a result of lost muscle fibers. Those muscle fibers just don't rebuild like they used to when we were younger. And less blood supply as a result of reduced density of capillaries, all those things contributing to, people when they're signing up for a running race or a biking race, you'll see that there's different age groups that people compete within, because we know a 50-year-old just can't compete with an 18-year-old even if they put the same amount of training in, because the muscles are different in an 18-year-old versus a 50-year-old. But this doesn't mean that as we age we're all doomed to being weak and frail and having reduced muscle mass, because the more active we are in our younger years, the stronger our muscles will be going into our older years. So we all know people that might be 80 years old that can live alone and walk to the grocery store and live a very active life. And then we also know 80-year-olds that are bound to the bed or the chair and very frail and weak and not able to participate in normal everyday activities. And a lot of that comes down to how active they were as young people. So it's really important for yourselves and people that you know to spread the word about the importance of activity. It strengthens our bones and it strengthens our muscles, and it'll make our older years much more enjoyable if we can continue to be active. So that concludes our lecture on the muscular system. I recommend that you again complete the homework assignment on the Mastering A&P website as practice for our lecture exam. Also, be sure to read the assigned pages that I put in the weekly learning plans for the muscular system, and then I'll help get you ready for our test.